Good day everyone, I'm Professor Chayokwans. We are from Group 20 and the group member is Laurasharwini, Sivakumar and me. Our article review topic today is the classifications of cancer types based on the gene expression data. Without any hesitations, let's go through the research. The overview of this research is to examine the accuracies of computer-based algorithms in cancer classifications and demonstrate the effectiveness of using molecular profiling in cancer classifications. In the studies, the algorithms involved include the support vector machines, frequency sets and associations rule, Chemists, and formal concepts analysis, whereas the types of the cancer evaluated are prostate adenocarcinoma, lung adenocarcinoma, breast cancer, kidney renal clear cell carcinoma, and colon adenocarcinoma.
So let's have a look at the methodology. Before we process the data has to undergo pre-processing before it follows the four methodologies of SVM, FCA, K-means, and also frequency sets and association rules. Lastly, it will undergo evaluation method where it will be evaluated for the most accurate method. So firstly, in data pre-processing, the data will be filtered and reduced using principal component analysis, where the real numbers were turned into binary codes for the algorithm association and class labels were converted into integers for the FCA algorithm. Then it was followed by the first method, which was SVM. For SVM, the main purpose is to create a hyperplane that will divide and classify the data according to the classes. So 50% of the data were used for training subset and another 50% were used for accuracy model subset. The second method was FCA, where complete classification was done using the data in a coding system. It classifies the objects with the same concept lattice from a dataset. The third method is K-means, where the dataset is divided into K clusters, resulting in high intra-cluster similarity and low inter-cluster similarities. The number of clusters is determined by the elbow method. The fourth method is frequency sets and association rules, where it classifies or associates data that are most frequently together. Data sets are divided into classes of different types of tumor based on gene expression data. After going through all these four methods, evaluation method is done, where the performance of the classification models or algorithm is evaluated according to accuracy. Based on the accuracy, it is determined by dividing the number of correct predictions for each of the method and with the total number of predictions. The most accurate method will be chosen for the data subsets. Now we'll look at the results for SVM. SVM was run on 3 different reduced data sets which were reduced before PCA and with 11 PCs and 5 PCs. The accuracy of each model was calculated at 99.8% with accuracy for 10 seconds of time run. The second was 99.2% of accuracy with less than 1 second time run, and the third was 94% accuracy with less than 1 second time run. So the most accurate SVM model was the third set of data set using 5 PCA, which gives an overall accuracy of 94% when calculated using the dataset confusion matrix. As shown here in the table 1, the confusion matrix for 5 PCAs are shown. The prediction values are shown according to the row, and also the precision values for each cancer class is shown from left to right, and the original values are shown in diagonal. So when accuracy value was calculated, the most high accuracy was shown for the SVM 5 PCA with the average value of 94%.
For the result of Chemists, the Chemists approach was applied to 2 datasets, which are the 11 PCA and 5 PCA. And in the figure below, we can observe that the elbow meters produce the ideal number of cluster of 5. The accuracy score for the 5 PCA dataset is 90.75%, while the accuracy score for the 11 PCA dataset is 91.75%, which is slightly higher than the previous one. For the FCA mediate, it was performed on 3 reduced datasets, which are 5 PCA, 11 PCA, and 5 genes using the univariate selections. The smaller datasets produce better results, with both 5 PCA and 5 US datasets producing approximately 83% accuracy, and both data have the identical accuracy, as opposed to the largest dataset, 11 PCA, which produce about 67% accuracy.
The last method used was frequency sets and association rules, and here are the 4 graphs for the 4 different types of sets. So the first is with the minimum support value, which increases from 0.4 to 0.8%, the accuracy of the model also increases from 17.25% to 46.25%. The minimum confidence was maintained at 0.6. Here, the minimum support was increased from 0.4 to 0.8, but the value of accuracy also was shown increasing from 31% to 49%. The minimum confidence here was also maintained at 0.6. So again here, if you look at 5 PCA and 11 PCA, when the minimum confidence value increased, the subsets remain unchanged. But here, when the minimum confidence value increases, again, the value of the dataset remains unchanged. So as such, using frequency sets and association rules, when we increase minimum support value, it provides a higher accuracy, especially when using 11 PCA's dataset. But when the minimum confidence value is increased for both 5 PCA and 11 PCA dataset, there is no change in the accuracy of the datasets. So as for the discussion, based on the table above, the method with the highest accuracy and shortest runtime is SVM at 99.2% when done using 11 PCA dataset. The hyperplane for SVM can be found if the dataset is divided according to the types of cancer pre to the processing of the data. The second highest accuracy is clustering the data with k-means on 11 PCA, which provides 91.75% accuracy, and 5 PCA dataset with the 90.75% of accuracy. The third most accurate result is from the FCA, which is Formal Concept Analysis, giving an accuracy of 93.1% of small dataset of 5 PCA. The frequent set and association rule model has a mid-range accuracy when the minimum support is increased but the minimum confidence is maintained. The highest accuracy is 72.25 when achieved using 11 US dataset with a minimum support of 0.8 and a minimum confidence of 0.6.
However, there are some limitations to the overall study as well as for each individual specific algorithms discovered during the classification process. One of the restrictions of a holistic analysis with cancer classifications is that some algorithms are able to classify data with high accuracies that only includes data from patients with a specific types of cancer, rather than including the patients without any form of cancer. For frequent set and association rule exhibit 3 limitations in the cancer classifications, one of which is that it cannot be used as classifications criterions because strong association rules may be not present in most tumor types. Second, it has small maximum size of frequent items, where the frequent sets are commonly found in various types of tumor and significantly reduce the accuracy of cancer classifications. Third, the a priori algorithms is computationally intensive and cannot perform calculations on datasets with a large number of attributes. And lastly, the limitations for the FCA is that the code system was originally designed for the image classifications. Besides, the major limitations is the issue with large datasets due to the way of the concept is built, where the number of the concepts grows exponentially as datasets increase, without the concept discover constraints. Lastly, these algorithms cannot perform on the reduced datasets with a larger species due to the system times and memory constraints if there is additionally precision information present.
As a conclusion, classification of gene expression data for cancer study can be done using 4 different types of method, which is support vector machine SVM, k-means, formal concept analysis known as FCA, and also frequent sets and association rules. The method with the highest accuracy is SVM when used with smaller dataset that has been previously divided into different classes of cancer. K-means provide the best second accuracy followed by FCA algorithm when again used with smaller datasets. The least accurate method is the frequent sets and association rules when used with the lowest minimum value. In terms of data, potential future research requires the dataset on both cancer and non-cancer patient to create a model that can predict the presence of cancer in a person. That's all from us. Thank you.